We're looking at one of the most impressive sets of eyes in the animal kingdom. And I do mean one of the most impressive sets of eyes because, let's be honest with ourselves. Anyway, this is the mantis shrimp, and they've received a lot of attention in the past couple of years because they have very unique sense of vision. Now, vision all comes down to one's ability to distinguish or to interpret different wavelengths of light. So humans have three types of what are called photoreceptors that allow us to see wavelengths of red, green, and blue. The mantis shrimp, on the other hand, has 12 varieties of photoreceptors, so they can see wavelengths of light that we cannot. However, humans can distinguish between more colors than a mantis shrimp. And the reason for that is the difference between reception and perception, which is the focus of our lesson today. Here we go. Perception of stimuli, section A.3. Look at this, right here. Got this fancy microphone today. Yeah, It's very fancy. This is a very fancy video. Hope you're ready. Okay, so first of all, perception. Let's start with the word reception, then we'll come back to perception. So reception is how your body can detect stimuli in the environment. We have photoreceptors, which can detect light or different wavelengths of light. We have mechanical receptors, which can receive signals for things like pressure. We have chemoreceptors, which can detect chemicals like smells in the air. Now, that's reception. Receptors are involved in reception, just detecting the presence or absence or concentration or quantity of a stimulus. Now, our brains have to interpret all of that sensory information, and now we're getting into the concept of perception. Perception is how our brains interpret or perceive all of these stimuli that our receptors are sending up to that big bundle of neurons up there. So we're going to take a few different examples of stimuli, how they are received in their receptors, and then how the brain will interpret those, and that gives us our perception of the world around us. So let's begin. I'm going to start in the skin, because your skin has several different types of receptors. It's actually very complicated. And so this gives us an opportunity to talk about how all of the different stimuli can be sent and then sort of integrated into a single thought up in your brain. So your skin. Now, we don't have to go crazy with excessive detail here, but it's important to note that there are specialized receptors embedded in different parts of your skin to pick up different types of stimuli. For example, you need to determine if something is hot or cold, so we have temperature receptors, which can receive stimuli regarding hot or cold, and importantly also pain. So those pain receptors are going to be very close to the surface of your skin, which is good because then your body can respond accordingly, because you don't want to freeze, burn, or die because you can't sense the pain involved in touching something. We also have receptors which are just dealing with touch, because humans rely on their hands to manipulate things like tools. It's part of our evolution, and so we need to have very sensitive touch receptors so we can manipulate things very carefully. Surgical instruments, paintbrushes, food. I do a lot of manipulating food these days. There's not much else to do. Stuck in quarantine, gotta eat. Then, in addition to those touch receptors, we can get more specific into varieties of touch receptors, and based on where they're located. I'm not going to go nuts here, but I do think that this one right here is rather interesting, because at your roots of your hair, you have to understand that your hair is actually part of your sense of perception of the world around you. Think of, I don't know if you've heard of how cats use their whiskers to basically understand where they are in space. It allows them to give them a sense of where they are in balance and coordinates their movements. And our hair does a similar function, actually. Your hair stands up when you're nervous or alerted to something, because it gives your skin more sensitivity, because as these hairs stand upright, they can be wiggled around, and then your receptors down here in your skin can pick up that wiggling, and it gives you a bit more sensitive touch. So anyway, your skin or your epidermis is loaded with all kinds of receptors. And that's about all we need to know about those. We're going to move on now to some more detailed stuff regarding your nose and how we can pick up odorants in the air. All right, we're looking at the inside of your mucous membranes, inside of your nasal cavity right here. So inside your nasal cavity, there are all these nerve endings. And if you recall from 9th grade when we did the brain dissection, there was this structure right on the frontal lobe, just hanging out under the frontal lobe. It's called the olfactory bulb. Here it is right here in yellow. Now that olfactory bulb is just a giant bundle of nerve endings. And then it has all these little extensions, these additional nerve endings that hang down through the bone and then out into your nasal cavity, just hanging there. And what they do is they detect different types of chemicals, which we call odorants. I'm going to take a closer look at those receptors now. So if we take a look at this diagram, ooh, here we go. Okay, this diagram here, this is the epithelium of the nasal cavity. And then these receptors, they just sort of dangle down through here. These are olfactory receptors. Now what's interesting is that we've evolved to have many different types of olfactory receptors, each one specifically designed to pick up one type of odorant. So, you've got your chocolate, not literally chocolate, but you've got your receptor which helps you determine just the smell of something like chocolate. Now, of course, it's not that specific. There are chemicals within the chocolate which tell your brain to either eat or disregard. It helps you determine which foods are safe or dangerous. But the important point is that each receptor is specifically designed to pick up just one type of odorant, which means we have to have tons of these receptors because we interact with tons of different odorants in our environment. Now, this is where we go from reception to perception, because just because you're detecting some kind of odorant in the environment, it doesn't necessarily mean that your brain needs to do something with that information. So the reception part here is that these odorants are detected by a receptor. Now, these neurons are going to fire, and they're going to send a signal up to the brain. Remember, the olfactory bulb is attached directly to the frontal lobe, and so it's a very quick response to that odorant. So you're going to pick up on that odorant. There's the reception part. That message gets sent up to your brain, and your brain has to interpret that odorant and decide whether this is something that you should be eating or this is something you should be avoiding. And so memories are very closely associated with odorants for good reason, because it keeps us safe, deciding which substances in our environment are going to be delicious and which ones are going to be deadly. So this is the very basics of how the reception of these odorants work. These are called chemoreceptors because they are designed to detect chemicals. So we had some pressure receptors in the skin, some pain receptors in the skin. Now we have some chemoreceptors up here in our nasal cavity picking up on odorants in our environment. So moving on to light reception, vision, in other words. There's a lot of detail in the syllabus about the anatomy, the physical anatomy of the human eye. For our distance learning edition of this section, we're going to skip a lot of that, and I'll just focus on the essential anatomy that allows you to pick up different wavelengths of light. So you do need to understand that your eye has this outer protective layer right here. It's thin, transparent, of course, outer protective layer. This is the cornea. Then the cornea, going beyond that, there's some fluid. Of course, we don't want air bubbles in here. We have some fluid. And then we move back from that fluid and we get to this colored region. This is where the pigment is located in your eye. This is your iris. And of course, you remember from the pupillary reflex that the iris can contract and dilate to regulate how much light gets back into the eye. That light is going to pass through the cornea, pass through the opening in the iris called the pupil, and then it must transfer through this structure called the lens. And it's just like the lens in a camera or a microscope, where it regulates the refraction of the light as it passes through into this empty chamber of the eye. So now the light has moved through the cornea, through the pupil, through this lens, and the lens can actually change its shape. It can kind of squish together or flatten out like this. And the way it does that is that there are muscles attached to the top and bottom which can yank on it. When it yanks on it, it brings the lens in a little bit tighter. And so that can change the focus, if you will, of the light moving through your eye, just like the lens on a camera would do. So as the light moves through the lens, it is focused, and then it gets scattered to the back of the eye where it makes contact with this region called your retina. And this is where we get into the detail of perception. I'm sorry, excuse me, reception. Oops, watch it, Lindenberg. Okay, so now we have receptors in the back of the eye on this region called the retina. And the retina is the only place in your eye where you're going to have a blood supply. Think about that for a second. Now, your eye, the whites of your eye, the cornea, you're not going to have a blood supply to that region. But you do need to have a fresh supply of blood going to the retina. Why? Because neurons require active transport, which requires ATP, which requires a fresh supply of blood with glucose so that we can keep those neurons functioning. So since there are neurons present, we're going to need fresh blood. So there's plenty of blood supply going to the back of the eye. That's the retina. So all of these photoreceptors that pick up the red, green, and blue light wavelengths are on the back of the eye here. And so what happens is the retina is going to basically just be loaded with these receptors, which we'll focus in on the next slide, which then can determine the type of light that's hitting your eye. And then that sends a signal to your brain, and your brain has to integrate all of those signals. There's a lot of red, there's a little bit of blue, and they have to assign a name to the color that we're seeing, and that's where the perception part comes in. So let's take 1 second, and we're going to take a closer look at the actual photoreceptors at the back of the eye, this region that we call the retina. Here we go. Okay, here we are looking at a close-up view of the retina. This is just the back of the eyeball. So the light has already passed through the pupil, through the lens, and it has moved through the empty space of the eyeball to the back wall, the retina. Here's just a small section of that retina. Notice the blood vessels, as promised. See, there are blood vessels here to keep those neurons fed. Now, as light hits the retina, it is going to basically interact with or hit these photoreceptors, which are located way back here. And they do that by traveling, actually the light passes through all of these layers of cells and gets all the way back to this region, which is pigmented. And this pigment in the back is going to absorb the light. Perhaps you'll recall from photosynthesis that any chlorophyll is a pigment, and the job of chlorophyll was to absorb light. And we have pigment in the back of our eyes, again, designed to absorb light. Now, those pigmented cells, of course, connect with receptors to different types of cells, which are neurons. And so we have a few different types of what are called photoreceptors that hook up with these pigmented epithelial cells. We have our rods and our cones, and I'm sure you've heard of these. The cones are involved in color vision. And again, like I said earlier, we have 3 different varieties of cones. That's the red, the green, and the blue. Now, those 3 types of cones will react differently depending on the wavelength of light that makes it to the back of the retina. So if you're looking at something that is carrying a particular wavelength of light, it could react with that particular type of cone. The rods are involved in monochromatic vision, and those are used for basically your sense of vision in dim light, where distinguishing color, is not important. You just need to be able to see. So when light is dim, you rely on your rods. This is kind of like your brightness, if you will, and the cones are involved in color. So we have 3 different types of cones, which pick up on 3 different wavelengths of light. And then, of course, we have our rods for monochromatic brightness. All of that information synapses with another type of neuron. These are called our bipolar cells, which then synapse with ganglion cells, and they all send this giant message down through the optic nerves, back through the back of the eye, and into the occipital lobe at the back of the brain. And then it's the job of the brain to interpret all of that information coming in from all of these different photoreceptors. Now, you might be hopefully wondering at this point, if we have 3 types of photoreceptors, how do we see all these different types of colors? And your brain must then interpret if you will, the quantity, how much red is present, how much green is present, how much blue is present, and we look at the ratios of those 3, and then your brain just makes up a color for it. And then we, as humans, make up a word in our language to describe that color. So we see yellow. We don't actually have a receptor for yellow. All our brain does is it looks at a certain concentration of red, blue, and green, and we just call that concentration yellow. And we see a different concentration of red, blue, and green, and we call that concentration orange. And then we agree as a culture on what orange and yellow and purple all look like. And they're all, again, just based on the ratios or the concentration of those 3 different receptors. And so the reception part here involves with the amount or the quantity of the wavelengths in the red, blue, and green spectrum. And then the perception part in the brain has to decide what those ratios actually mean. So that's how vision works in the human brain, relying on the reception from photoreceptors and then the interpretation of what that actually means to call it a color, like yellow or green or blue. We're going to wrap up with 2 more types of reception, which both involve the ear. That's sound reception and then your spatial sense. Both of those are determined by your ear. We're going to start with sound reception. And the way this works is that your sound is a physical, mechanical process, because all we're doing is we're just trying to interpret the size of the vibrations moving through the air. So if you imagine someone clapping their hands, that clap is going to send little vibrations through the air. It's going to disturb the molecules in the air in a wave. Now, those waves or those vibrations will eventually land at your ear. This is the outer ear, called the pinna. And the pinna is basically just like a cone, which helps to funnel all of those vibrations into this region right here, which moves us into our inner ear. And so those vibrations bounce around in this little tunnel called the auditory canal until they land on this little flap of tissue called the eardrum. You've heard of this, also called the tympanic membrane. Timpani, think of the big drums in an orchestra, if that means anything to you. Timpani, tympanic membrane, it's the eardrum. Now the tympanic membrane, or the eardrum, is going to absorb those vibrations, and it's going to shake just a little bit due to the vibrations. Those vibrations are going to be transferred to 3 little tiny bones. These are in fact the smallest bones in your body. These are called the ossicles collectively, and the ossicles are going to transfer those vibrations to this structure, the snail-like structure. This is called the cochlea, and this in the cochlea is where the actual receptors are located. So all of that mechanical movement of vibration in the air gets sent through the auditory canal, transferred from the eardrum to the ossicles, and then bumps up against the cochlea. And the cochlea is a rather complicated organ or structure. What the cochlea does is it's a tough, it looks like a snail shell, and in fact it feels like a snail shell because it's a rather tough bone-like structure filled with fluid. And the fluid on the inside is going to pick up those vibrations coming in from these ossicles. So these ossicles vibrate against a little membrane here on the cochlea called the oval window. Now those vibrations transfer through the cochlea in this snail-like structure, the spiral, and they just bounce against the walls all through the cochlea. And as those vibrations are moving through the cochlea, they're going to bump against little structures called hair cells. I'm going to zoom in on some hair cells here. So here's your cochlea again. Let me get out of here. There we go. So those vibrations are going to be moving through the inside of the cochlea. As they do that, you see these little finger-like projections? Those are the hair cells. Now those vibrations bump against those hair cells, and that bumping causes those hair cells to move just a little bit. Now it's important to note that when vibrations are moving through the cochlea, they're not all the same size. We have to talk a little bit about physics here, but waves have different sizes, right? So the wavelength, that space between the waves, can be different, and the different wavelengths could be very, very short like this, or they could be rather longer like this. And so those wavelengths will affect these hair cells differently. And so what your cochlea is designed to do is to have multiple different types of hair cells like this. And those hair cells respond only to very specific types of wavelengths. So if you have a very big wavelength, think of like a bass. You have lots of bass coming at your ears. There's lots of bass in your face. It's moving into your ears right now. You'll have very specific hair cells which pick up low frequencies for those low bass wavelengths. Now imagine something very high. Those are going to be moving more quickly, and those bass hair cells might not pick up those frequencies. You'll have different hair cells which pick up those higher frequencies. And so then you have a collection of different types of hair cells which all send, again, signals to the brain, and your brain has to interpret all of those high and low frequencies and determine different types of sounds. Now what happens, and you can see it in this picture here, is that certain frequencies can hyperstimulate these hair cells, and they can actually die as a result. Perhaps your parents have warned you not to go to concerts because you can kill your hearing. And that's actually true, because at concerts what happens is it's so loud that the low frequencies, this is important, the low frequencies, not the high frequencies, but the low frequencies with their longer wavelengths can actually pummel these hair cells and kill them. And you can see right here in this person's cochlea, this is a loss of nerve fibers, a loss of hair cells, because they were, I don't know, hyperstimulated, perhaps because of too much bass, which can kill those hair cells. In addition, as you age, and this is already happening to me, but as you age, the hair cells involved with high frequencies, they naturally just die out. For whatever reason, those are the ones that die out first. So you've heard that there are certain frequencies that older people cannot hear. That's absolutely true. And the reason that it's true is that these high frequency hair cells die sooner than the low frequency hair cells. I don't know exactly why, but that's how it works. So you start to lose the hair cells which are specifically designed for high frequencies first, and then later in life you lose more and more and more until your hearing's almost completely gone. And so that is that's the basics of how these different wavelengths are interpreted by the hair cells. Now, what happens after the hair cells are stimulated is that all of those hair cells send a signal to this giant nerve called the auditory nerve, carrying all the information from all the different types of hair cells up to the brain. To the brain. And your brain is going to interpret the meaning of the different wavelengths of sound that they hear. Excuse me. And then we go from the reception of vibrations to the perception of sound or language or music in the brain. And so that's our final receptor as far as the outside world goes. The last one, still involving the brain, excuse me, not this slide. Here's our cochlea again. The last type of receptor is located in the cochlea, and these extensions up here, here's one, here's another, and here's another. Now these extensions are what we call the semicircular canals. And the semicircular canals are filled with fluid, just like the cochlea. But they're designed to react differently to the fluid. Now these semicircular canals are designed to pick up movement of the fluid. So if you turn your head this way, then the fluid in the semicircular canals is going to start to swish in that direction due to momentum and inertia. Now, as that fluid flows through those semicircular canals, it's going to flow across some receptors. It's going to physically push them over. So they're standing upright, the fluid flows against it because your head turned, and then those little receptors start to bend. And as they bend, they stimulate a nerve which sends a signal to your brain that says, oh, your head is moved this way. And so it helps your eyes to readjust quickly to the movement in your head. If you look up, a similar reaction will occur. So we have semicircular canals moving out into different planes in space so that you can pick up on different movements of your head. This allows you to determine what's going on around you, even if your head is, sideways or upside down, because the semicircular canals allow your brain to reorient itself by picking up on movements of the head. Incidentally, if you spin around in circles, you will get dizzy, because what happens is that fluid continues to spin even after you stop moving. So the inertia of the spinning keeps that fluid moving even after you stop. And then your brain thinks that your head is still spinning around, even though you've stopped moving. So that fluid moves, your brain thinks that your head is moving, and so your vision gets all screwed up because the fluid keeps moving through those semicircular canals, hyperstimulating these receptors, and your brain thinks it's still spinning, and so your vision gets completely messed up, and that's how you feel dizzy after moving around. Here's a close-up view of those receptors in the semicircular canals. So what happens is the fluid moves through that canal, and it's going to basically, knock this little receptor over. So as this thing tips to the side, it stimulates these nerve cells, and then it tells your brain that you have tipped or spun your head around a bit. That's the end. We've reached the end of perception of stimuli. And this brings us back to the mantis shrimp. Even with its incredible set of 12 photoreceptors, even with these very unique eyes, the mantis shrimp cannot distinguish between colors that the human eyes can distinguish with just our 3 varieties of photoreceptors. And this all comes back to perception. While the mantis shrimp can certainly out-hunt us at the bottom of the ocean, our eyes will win out every time when it comes to distinguishing between colors. And that's not because of the eyeballs themselves. Rather, it has more to do with our brain's ability to integrate and interpret information. In other words, our perception of the world around us perhaps has made us more successful than the reception of the world around us. I hope you found this content interesting. I love it. Your assignment is posted to Teams. Make sure you get that into me by the end of the week. And you guys have a good week. Take care. Talk to you soon.